Hello friends. Today the topic which I am going to discuss with you is about antibodies. This is one of the most important topic these days, as number of diseases are happening around us or we are suffering from them. And these are the structures which help in providing immunity to our body. When we say the word immunity, that means defense system of the body. So what we can say in easy words, we have soldiers inside our body which fight against the disease causing organisms or microorganisms. Now, these antibodies are also regarded as immunoglobulins. They are also named as immunoglobulins. There is a common notion which says all antibodies are immunoglobulins, but all immunoglobulins are not antibodies. Reason being, there are many immunoglobulins which perform other functions of the body and they are not acting like antibodies. The word immunoglobulin can be divided into two categories. The term amino means which provide immune system to the body. And globulin is basically a protein structure. So there are number of proteins are present in our body, and out of that, one protein is named as globulin protein. And this protein help in providing immunity to the body. Now, these antibodies or immunoglobulins, we have given them the abbreviation IG, capital I, small g, capital A, small b. These are the abbreviative terms of the antibodies and immunoglobulins. Now, where you find these antibodies or immunoglobulins in your body? These structures are found in the serum of the body. Serum which become a part from the plasma. Now this thing was confirmed by two scientists, Tiselius and Cabot. These are the two scientists who confirmed that antibodies or immunoglobulins found in the serum of the body, and their structure was explained by Porter and Edelman. These two scientists have explained their structure in detail. In order to explain their structure, they use two different enzymes. Which are named as papain and pepsin. Papain and pepsin. They used both of these enzymes to digest the antibodies. When they digest it with papain, two structures are formed of the antibody. One is called as FAB, fragment of antibody, and the other one is called as FC, fragment of crystallization. Fragment of crystallization. When we do cooling of the structure, they get crystallized. And they play the role of antibody, that means which fight against the antigen. Now this pepsin is a very well known enzyme in our gastric juice, and it is a proteolytic enzyme. This enzyme digests again antibody into fragment of antibody and other number of fragments. Other part, number of fragments. As this papain enzyme is not specific in digestion, so that's why it forms only 2 fragments, whereas pepsin enzyme form a fragment of antibody and number of fragments. From here, with the digestion of these enzymes, they confirm the structure of antibodies or immunoglobulins. Now, when we talk about the structure, of antibody, then we say, here we will write the structure of immunoglobulins or antibodies. Then we say, structurally, they are formed of 2 different types of chains. 2 different types of chains. And those chains are named as, light chain and heavy chain. As their name depict, light means they are light in molecular weight. Heavy means they are heavy in their molecular weight. Now before that, I just want to discuss a little bit about antibodies. That is the chemical nature of these antibodies or immunoglobulins. These are glycoprotein molecules. When we say the word glycoprotein molecules, that means it has 2 components. One is carbohydrate and other is protein. So antibodies is a composite structure and they are formed of 2 components, carbohydrate with association with the proteins. That's why we use the term glycoprotein. Now here in this structure, again we come back to our original topic, structure, heavy chain and light chain. These chains are basically what? These chains are basically polypeptide chains. That is the chains with number of different amino acids. And I told you, light chain means they are light in molecular weight. They have less molecular weight because their number of amino acids are less as compared to heavy chain, which is having higher number of amino acids. Now, each chain further, once again, whether it is a light chain or a heavy chain, each chain, we can write over here, each chain further has 2 types of components. That is variable and constant. Variable and constant. In majority of antibodies or immunoglobulins, we just have one variable region. But constant regions, they can vary from 3 to 5 in number. 1 in number, 3 to 5 in number, depending upon antibody to antibody. That we will discuss shortly now. Now, each chain, again, each chain further has 2 terminals. Each chain further has 2 terminals. That is amino terminal and carboxyl terminal. Amino terminal, that is the chain ending into NH2 group, and the chain which is ending into COOH group. They are accordingly called as amino terminal and carboxyl terminal. Why they have amino and carboxyl terminal? Because I told you that they are made up of polypeptide chains. And polypeptide chains are made up of beta what, please? amino acids. They are made up of amino acids. And each amino acid having at one end amino group and other end having carboxyl group. Something like this. This is the structural formula of amino acid. So when they join, at one end amino terminal left, on the other end carboxyl terminal left. Now structurally, these antibodies or immunoglobulins, they resemble with the English letter, their structure resemble with the English letter capital Y. Their structure resembles with the English letter capital Y. Now, we will draw this diagrammatic structure. How many light chains are there? 2 light chains are there. How many heavy chains are there? 2 heavy chains are there. Now if I draw the diagram, Here you can see, I have just drawn the outline of the antibody structure. We can extend it a little bit more in order to show all the structures in a good manner, so that all the structures which are present associated with it, they should depict clearly. We can see them clearly. Something like this. Something like this. So diagrammatic representation of the antibody resembles like this. Now, what is this? This is light chain. You can see it is smaller in size than the other chain, which is named as heavy chain. Light chain and heavy chain. 2 light chains and 2 heavy chains are present associated. All these chains are interlinked with the help of bond, and that bond is called as disulfide bond. Those bonds are called as disulfide bond. These bonds are present between the heavy and chains, also between the heavy and light chain. They connect the same chain and different chains together. Something like this. Disulfide bond. This is how they are connected with each other. Now each chain, I told you further, consists of variable and constant region. Now this side is variable light. This is variable heavy, and this is constant light. Similarly over here also, variable light, constant light. Light means light chain. Similarly over here, variable heavy chain, constant heavy chain, constant heavy chain, H1, because they have more than one constant regions. So that's why I have mentioned CH1. Then we can write CH2, CH2. Then we can write CH3, CH3. constant heavy second, constant heavy third. So they have different constant regions. But variable region, whether it is light or heavy, they are 1 in number. Constant region in case of light chain is 1 in number. But constant region in case of heavy chain, they are more than 1. So this is how we draw the diagrammatic structure. Now, as I told you, they are also composed of carbohydrate. These carbohydrate molecules are present attached over here. These carbohydrate molecules are present attached with the antibodies over here, with the heavy chain. Next, each terminal, this side, this is amino terminal. This is amino terminal. This is carboxyl terminal of the light chain. Carboxyl terminal of the light chain. This is the amino terminal. This is the amino terminal of the light and heavy chains both. And these are the carboxyl. These are the carboxyl terminal of the heavy chains. Now, further, at this point, each antibody can bind with the antigen. Now, the place where antibodies bind with the antigens, that place is called as antigen binding site or hypervariable regions. These are hypervariable regions. That means each antibody has two antigen binding sites. And these sites are different for each and every antibody, depending upon the antigen. Look, just imagine this is an antigen. This is an antigen. On the antigen, whole of the antigen never binds with the antibody. There are specific locations on the antigen. There are specific locations on the antigens where antigen can bind with antibody or antibody can bind with antigen. These locations are called as epitopes. These locations are called as epitopes. So these are the structures where antigen is going to bind with the antigen binding site or hypervariable region of the antibody. Very, very important. Why this region is called as a hypervariable region? As I told you, this region is having huge amino acid differences. That's why it is variable. Those amino acid differences make the antibody to bind with the different types of epitopes of the antigen. That's why hypervariability. Now, that's it. Now, further these immunoglobulins, further have been categorized. They have been categorized into different categories. Here I write type. When the scientists, they discovered the different types of antibodies in the human body or other organism body, they discovered it following types. The first type is IgG. I am going to mention all those antibodies which have been discovered according to the percentage from maximum to minimum. Then IgA, then IgM, then IgG, E, and then in the last we have IgD. So that means 1, 2, 3, 4 and 5. 5 different types of antibodies have been discovered till today. Now on what basis we have categorized them into 5 different types? That is a very, very important question. Look. They have light chains. Till today, only 2 different types of light chain have been discovered. They may be kappa or lambda. So that means kappa or lambda, kappa or lambda, kappa or lambda, kappa or lambda. That means on the basis of the light chain, we can't categorize the antibodies into 5 types because they themselves only 2 types. Then comes heavy chain. Heavy chains on the basis of which these antibodies have been categorized. In case of G, heavy chain type is gamma. In case of A, it is alpha. In case of M, it is mu. In case of E, it is eta. And in case of B, it is delta. That means these antibodies have been categorized into 5 different categories on the basis of type of heavy chain they possess. For example, in this diagram, if I mention this heavy chain as gamma, that means this immunoglobulin is IgG. But if I write over here delta, that means this immunoglobulin is IgD. I think you understand. So depending upon the type of heavy chain, antibodies have been classified into 5 different categories. Now, percentage in the serum, how much percentage they are present in the serum. Maximum is IgG, that is 80%. Then IgA, 10 to 12%. Then IgM, 5%. Then E, it is less than 1%. And IgD, it is about 0.3%. So that I have told you, I have categorized according to the percentage. I have mentioned their types according to the percentage in which they are present in the serum. And type of antibody depends upon the type of heavy chain they possess. Next. What functions they perform specifically? If they are different, that means they are performing different functions too. Very, very important functions they have. Like IgG can cross placenta. If a woman is pregnant, that woman is providing immunity to its embryo through the antibody IgG, because IgG, antibody of the mother, can cross placenta and provide immunity to the embryo. It activates complement system of the immune system. Complement system. It's a system of proteins, where one protein complement the second complement the third, in order to start the immune reaction. Around 13 different complement proteins are there to start the immune reaction. Then most abundant most abundant antibody during secondary immune reaction. Not in primary. I'm not saying primary. It is most abundant during the secondary immune reaction, when the same antigen enter into your body for the second time. Or you can say when your body encounter the same antigen for the second time. At that time, your body has memory cells and they start fighting. That immune reaction is faster. At that time, IgG is in more amount, maximum amount than any other antibody. Then next, it has been categorized into further IgG1, IgG2, IgG3 and IgG4. 4 different IgG immunoglobulins have been discovered. Then comes IgA. This antibody IgA is specifically named as secretory antibody. Why it is regarded as secretory antibody? Because it is present in all the secretions of the body, like saliva, like tears, like sebum. All these secretions of the body, human body, they contain IgA antibody. And the most important, which I have not mentioned yet, it is present in the colostrum. Colostrum is the first milk produced by the lady after the delivery of the child. So the first milk produced by the lady after the parturition, that milk is called as colostrum, and it contains IgA antibody, which is very, very good for the growth of the child. That's why doctors nowadays recommend mother feed should be essential for the growing child. It has also been classified into 2 subcategories, IgA1 and IgA2. One thing very more important about this antibody is it exists in dimeric condition. That I will discuss little later. Next is IgM. IgM, very important. It is the largest antibody and it exists in pentameric condition. Pentameric condition. That means, as I have written over here, dimeric, Ig exists in dimeric and M exists in pentameric. That means dimeric means 2 such structures they will join together. 2 such structures they will join together, something like this. Something like this. Something like this. With the help of a chain, that chain is called as a J chain. With the help of a chain which is called as J chain. In case of pentameric, these molecules are present something like this. Pentameric. So this is a IgM antibody. That's why it is the largest antibody. Pentameric condition. And it is produced in primary. It is produced in primary immune reaction. So whenever you encounter an antigen into your body, this is the first antibody. This is the first antibody produced against that particular antigen to provide you immunity. Very, very important. So that is called as primary immune reaction. Also, neonatal, the child which has taken birth, that is neonatal will synthesize this antibody as his own first antibody. Neonatal, the newly born child the first antibody synthesized by him or her is IgM. So that means it has a big importance for the neonatals as well in primary immune action. And as I told you, it is a pentameric in structure. So already told you one antibody can bind with 2 antigens. Here you can see 10 antigen binding sites are there on the IgM molecule. But out of 10 antigen binding site, it is able to bind with 5 antigens only because of steric hindrance. There is a steric hindrance. Now, next is IgE, fourth antibody. Light chains may be kappa or lambda, and its heavy chain is eta, on the basis of which they have been categorized. It is less than 1%, and this antibody is called as allergic antibody. And it is produced or present on the surface of mast cells or basophil cells in the body. They play a great role in allergies like hay fever, anaphylactic shock, asthma. These antibodies are produced during those allergic reactions. In the last, we have IgD, which is the light chain is kappa or lambda, heavy chain is delta, and they're present less than 0.3%. And though their function has not been properly identified, but it is said they play a role as anti-cancerous antibody. They play a role in providing immunity against the cancer. So this is how we have discussed in detail the structure of all the antibodies and how they are different from each other, how they are different from their functions, structure. That is all about today's lecture. Thank you.